Assalamualaikum, my name is Nur Akila. Today I will give some explanation for the cyanogenic glycoside. What is cyanogenic glycoside? Cyanogenic glycoside is a natural plant toxin that found in a few plants. It is a synthetic mixture contained in food sources that discharge hydrogen cyanide when beaten or processed. Cyanogenic glycosides are common in certain families such as Fabaceae, Rosaceae, Leguminosae, Linaceae, and Compositae. There are approximately 25 known cyanogenic glycosides, and these are generally found in the edible part of plants such as apple, apricot, cherries, peach, plum, quince, and particularly in the seed of such fruit. The chemicals are also found in almond, stone fruit, palm fruit, cassava, bamboo shoot, linseed, lima bean, cocoyam, chickpeas, and cashews. Next slide is for the example of cyanogenic plant food. The first one is cassava. Cassava contains high amount of cyanogenic glycoside. Cassava is also a staple food in most African communities. Next plant is cocoyam. Cocoyam can be converted into flour and used to make mash meal or porridge. Cocoyam also contains other sources of nutrients such as protein, vitamin, carotenoid, and mineral. Apart from the nutrient composition of cocoyam tuber, the crops have been reported to contain low level of cyanogenic glycosides. Next is bamboo shoot. Bamboo shoot contain lethal concentration of cyanogenic glycoside. The cyanogenic glycoside present in bamboo shoot is taxiphyllene, which is decomposed quickly in boiling water. Although cyanide content of bamboo shoot is much higher than that of cassava root, the cyanide content in bamboo shoot decreased substantially following harvesting and processing. Next is sorghum. Durin is the major cyanogenic glycoside in sorghum. The cyanide content of sorghum increased rapidly during early growth of stage, after which it declines with plant age. The most common cyanogenic glycoside in fruit and fruit kernel is amygdalin. Amygdalin content of the fruit seed varies significantly from seed to seed. For apple can be consumed raw or processed. Although apple are rich sources of vitamins and other nutrients, apple seed contain high level of cyanogenic glycosides. Next slide is regarding the health implication of cyanogenic glycoside. The toxicity of cyanogenic glycoside and the derivative is dependent on the release of the hydrogen cyanide. The toxicity may result in acute cyanide poisoning and has also been implicated in the etiology of several chronic diseases. The dietary exposure to elevated level of some cyanogenic glycoside in food has the potential to cause acute cyanide poisoning or a debilitating, irreversible neurological condition in the long term. High and sustained cyanogenic intake may result to disease such as Konzo. Konzo is an upper motor neuron disease characterized by irreversible but non-progressive symmetric spastic paraparesis that has an abrupt onset. It mostly affects children and women of childbearing age. Next is tropical ataxic neuropathy (TAN), which is used to describe several neurological syndromes attributed to toxic nutritional causes. Goiter and cretinism. This disease is particularly common in Africa because of their overdependence on cassava as a staple food. The continuous exposure to dietary cyanide from insufficiently processed cassava products aggravates the disease by the interference of thiocyanate with the dietary iodine, thus leading to iodine deficiency. Next is growth retardation. The exposure to cyanogenic glycoside has been a contributing factor to this health problem. Growth retardation is particularly a serious problem in population consuming food with inadequate protein, especially diets that are low in sulfur containing amino groups. This is because detoxification of cyanide in human body requires sulfur donors from the sulfur-containing amino acid. Thus, dietary exposure to cyanide is a contributing factor to growth retardation. Next is cyanide poisoning. Cyanide poisoning occurs as a result of consumption of bitter cassava, almond kernel or apricot kernel and their product without proper processing. Cases of cyanide poisoning after consumption of drink produced from the blend of apricot kernel and orange juice have been reported. Clinical symptoms of cyanide toxicity are vomiting, nausea, dizziness, pain, stomach pain, weakness, headache, diarrhea, and occasionally death. In conclusion, cyanogenic glycosides are abundant in edible plant. Consumption of improperly processed cyanogenic plant can lead to chronic and acute heart problems. Understanding the appropriate processing method for specific cyanogenic plant will help in reducing the problem of unintentional cyanide toxicity. Similarly, to prevent adverse effects of cyanogenic glycoside in food plant, consumers should prepare food properly before consumption. Explain the structure of cyanogenic glycoside. Cyanogenic glycoside have consisted alpha hydroxynitrile glycone and a sugar moiety that producing hydrogen cyanide during the hydrolysis process. This hydrogen cyanide producing property underline the toxicity of the plant commonly associated with this compound in human and animal. However, new evidence shows that the toxicity of this compound is lower than those theoretically caused by the equivalent release of hydrogen cyanide. For the next question, how does cyanide detoxify in human body? Cyanide is detoxified in the body by the enzyme rhodonase with the help of sulfur-containing amino acid to thiocyanide, which is excreted in the urine. However, the detoxification mechanism of cyanide in the human body can only cope with low level of cyanide generated from consumption of small amount of cyanogenic plant.